this diagram here illustrates that there are a number of drugs that are in development or already exist out there that target various elements of the pathway that lead to antibody-mediated rejection, including activation and maturation of these cells, as well as targeting plasma cells, antibodies, and complements. So there's a lot of work that needs to be done in testing these drugs and in trying to find which are the most effective. And we also need to be able to compare them. And that's just to make the point that it's really important that we have a certain level of agreement on what disease entity we're trying to treat and how we're going to define it. So this is just a really brief overview of what I'm going to cover. So antibody-mediated rejection either as an enrollment criteria or as an endpoint. And very briefly, our role as pathologists sometimes can be that we are looking at biopsies from patients, we make a diagnosis of AMR, and then that might lead to their enrollment in a clinical trial. Another role that we might play is if patients are enrolled in clinical trials, they might then have biopsies taken as part of their follow-up protocol biopsies or indication biopsies, and we have to look at those as well and look for criteria, features of antibody-mediated rejection. And those trials can be either trials to prevent the development of AMR, so we're trialing a new treatment against standard of care, and then we're following up with patients to see what effect that has on their GFR or graft loss. And very often in these trials, the diagnosis of AMR and the biopsy is really crucial to the endpoint, given that that's what we're trying to prevent. And then on the right-hand side, you have trials of treatment of rejection, in which patients enrolled based on the diagnosis that we might make, randomized to two arms. This is just supposing a very simple clinical trial. And then in these trials, very often actually EGFR and graft loss are the important outcome, but there might be protocol biopsies built in, or there might be indication biopsies that are part of the endpoint or a secondary endpoint. And on that note, I just want to also clarify the definitions of endpoint, primary, secondary, and surrogate. And I won't be talking about surrogate because we have talks on that later. So the important point that I've highlighted here is that the endpoint's obviously a variable that's intended to reflect an outcome of interest, and it must be precisely defined in order to yield reliable and reproducible results. The primary endpoint is particularly important because that's the one that's used for the statistical design, the power calculation to determine the sample size for the trial. And that endpoint can be composite, and often is, so that if you're looking, for example, at a trial of prevention of antibody-mediated rejection, you're not just looking at AMR as a diagnosis in the biopsy, you're also combining it with EGFR and graft loss. And then secondary endpoints we allowed a little bit more flexibility with because they don't have the same statistical authority, and that's often the case, as I showed you in the previous slide, for trials of treatment of AMR, where your primary endpoint is functional graft loss, but biopsy features might also be considered as a secondary endpoint. So both the FDA and the EMA consider biopsy-proven rejection as an important primary endpoint in transplantation, either in its own right or as a composite endpoint with other features. So that's one good thing. But that supposes biopsy-proven rejection, not necessarily distinguishing between T-cell and antibody-mediated rejection. And so an important question here is, should we use AMR as an endpoint in its own right rather than as part of a sort of blanket biopsy-proven acute rejection? So let's have a look now at how we might define antibody-mediated rejection for clinical trials. And this is just to remind everyone in the audience that right from its inception, the BAMF classification was designed to be used not only for diagnosis in individual patients, but also to allow to have these objective endpoints for clinical trials. And that brings us back a bit to some of the discussion we were having with Alex earlier about what is the purpose of the BAMF classification. Is it just for diagnosis? Is it for prognosis? Is it for clinical trials? These are all slightly different needs. And so to go really briefly through the evolution of the diagnosis of AMR, which I'm sure you're all familiar with, very presciently again, these 6 categories that we have now are pretty much unchanged, and the second one was already AMR at that time, although the only variant that was recognized as this form of hyperacute rejection due to preformed antibodies. And over the next decade or so, that was extended to include delayed forms, so accelerated acute antibody-mediated rejection. And then with really important progress in 2001 with the use of C4D and higher sensitivity assays for DSAs, that was expanded. But still at this time, these criteria were very much focused on these sort of early post-transplant AMRs, quite acute in patients who are often sensitized or, with preformed antibodies. Chronicity was introduced in 2005, and it's not really until quite a bit later in 2013 that we ended up extending this AMR category to include more chronic lesions, so the sort of lesions that you see more often with de novo antibodies late post-transplantation. And you can see at this time a number of changes were made, introducing C4D negative AMR, introducing endarteritis and multi-chronic angiopathy, EM features of chronicity, the molecular signature, and non-HLA antibodies. So I think where we're at now with the 2019 definition is that we've developed a very good classification diagnostically that aims to capture all patients that have or might have antibody-mediated rejection. But because of that, it's become very heterogeneous. Many speakers have mentioned this before, in terms of early and late AMR, in terms of the degrees of activity, chronicity, complement activation or not, molecular features. And we now know that probably in this basket, along with the sort of early acute AMRs that we started with, and then the later AMRs from the 2000s, we now have some things that may not actually even be related to antibody. And then the other point that I want to make is that there's some problematic elements in this definition. These are things that I find hard to work with as a sort of in my daily practice as a pathologist. So how do we define acute tubular injury, which is one of the features of active AMR? How do we rule out all other causes when we use TMA for the diagnosis of AMR? Same thing for V lesions. And how do we establish new onset CV lesions, for example? And then there are a number of features that aren't routinely used in most labs, EM and molecular analysis. So having set that scene, let's just have a look at how previous clinical trials to date have used the definition of AMR. And I've separated it into 4 categories: prevention, treatment of AMR, and then early and late, because they're quite different scenarios. And I'm going to show you a table like this for each one, which has quite a lot of data, but I'm not going to go through it in detail. What I want to focus on really is the purpose of this talk. It's the AMR definition that's been used. And so if you look at trials for the prevention of early AMR in sensitized patients, these are all trials from the last decade or so. And you can see in the endpoint type that biopsy-proven AMR is a really important primary endpoint, sometimes used also as a secondary endpoint when looking later post-transplantation for chronicity, for example. But the definition used often goes back to that early definition, which makes sense given that that definition was sort of constructed around these early AMR cases. So even though they're quite recent, they're using older definitions of antibody-mediated rejection. When we look at prevention of late de novo DSA, there aren't that many studies out there because the timelines are obviously huge. And in this situation, again, unsurprisingly, I don't know why it's coming out white, but that is biopsy-proven AMR on the bottom. Unsurprisingly, that's often used as a secondary endpoint, so the endpoints are much more often related to creatinine and graft loss. And here, again, makes sense that the definitions used are a little bit more recent, although they're not the most recent. These are 2017, 2020 papers, so the clinical trial must have been conducted over the 5 to 10 previous years. So they're still slightly using slightly older definitions. Not many interventional studies for the treatment of early AMR, but I'm just showing you this one here, in which AMR is used as a primary composite endpoint, but not using the exact BAP definition, so using an expanded AMR categorical scorecard. And then lots of trials for the treatment of late active chronic active AMR, where often the AMR endpoint, if you look on the right-hand side, is a secondary endpoint. And if we also, it's interesting to look at which patients were enrolled in these trials of treatment of later active or chronic active AMR. It's often not using the full BAMF criteria. So some use BAMF 2013, 2007, 2017, use that to enroll patients. But other trials will use a sort of mixture of BAMF lesion scores, DSA, not DSA, and often actually the molecular microscope as part of the enrollment criteria. So what I conclude from this is that firstly, trials have been using AMR in its own right to enroll and as an endpoint, but they've not been using the BAMF strict definitions. They've been using adapted definitions to their clinical query and or BAMF lesion scores. And so although it was very much part of the purpose of the BAMF classification at the beginning, the BAMF definitions may not be fit for purpose now for their use in clinical trials. And then we've heard from previous speakers also early in the AMR are clearly different. Can we use the same definitions for those two in clinical trials? So as part of this ESOT initiative, which was led by Martin Nassens, I helped out with working group 1 up there, which is histological endpoints. And we wanted to propose some updated definitions of rejection's primary endpoints. And in particular for antibody-mediated rejection, this is what we came up with, which was firstly simply to recommend using it obviously as an endpoint in its own right, which is what everyone's been doing anyway, but also to suggest that some elements were not really suitable for to be included in definitions as endpoints because of their lack of reliability, and also because they're not as frequent. So we were proposing that the most reliable features of AMR, the most frequent, the most correlated with outcome, are microvascular inflammation scores of 2 or more, and in terms of chronicity, having double contours on light microscopy and having them at a higher threshold that's more often associated with sort of established chronicity, so 10% of capillary walls. And we also acknowledge at this stage that the definition may have to be different depending on whether you're doing a trial for early or late AMR. And then we also included in this the BAMF recommendations of best practice for histological molecular endpoints in clinical trials that were laid out in the BAMF 2017 report. And emphasizing again that because endpoints need to be reliable and reproducible, there are a number of steps that we can take to ensure that, including such as including pathologists in the design, and also several pathologists for the scoring and an adjudication mechanism, granular data, digital pathology, etc. And I suppose what I'd like to bring to this audience is whether, as a community, we can, or not, take those recommendations a bit further. And I've listed the questions that I think I'd like to discuss with you. Is the first one, having AMR as an endpoint in its own right, can we endorse that? Can we agree on the definition of antibody-mediated rejection for clinical trials? And what should we use then? So BAMF 2019 or simplified version, different for early, different for late, different for enrollment or as an endpoint. And sort of separately and not covered by this talk, also whether the data that we use in our clinical trials, we should endorse some kind of statement, we should put out some kind of statement saying that the sort of granular pathology molecular data should, if possible, be made available for systematic review so that we can learn from the different trials that are carried out together. So I think I'll just leave those up. That helps. That's the end of my talk.
Thank you, Candice. So let's start and try to discuss.
Yeah, unless anyone has done.
So I struggled with the definition. We've got this trial with an R6 blocker that we've had trouble recruiting because the definition is really, really tight. And so, for instance, if it's a late, I mean, most of ours are late, non-compliant, they'll have mixed rejection. There'll be interstitial infiltration. that automatically cuts out the MVI. So automatically you then lose your PTC, and unless you've got G, and we don't see that much G in it. If they've had a pulse of steroid before they have the biopsy, we don't see it, in which case then they might have, say, transient glomerulopathy, they might have a stonking antibody, 5,000, 10,000, but we can't call it chronic antibody mediated rejection because it doesn't fill all the criteria and I can't recruit them.
Yeah, I think that's a problem that many people have in antibody-mediated rejection. I think that's one of the problems with the definitions as they stand, is that they're designed for diagnosis, and when actually looking at a clinical trial, yeah, you keep on having to not know people. I think that's a big problem.
Can you tell the difference between a PTC that's involved in antibody versus a PTC involved in just a low-level, because quite often they'll have a low-level TCMR as well. There'll be an incision portrayed with these late biopsies.
Well, we've seen with some of the molecular data that was presented on Monday that PTC is the strongest correlate with the molecular signature of AMR.
Yes.
So maybe that's something that molecules can answer, or maybe it's something Nicholas Kozakowski can answer.
Yes, this is something that we would like to instate within the path for approval. We are gathering cases to respond this question.
But it's reasonable to have different definitions of ABMRs.
I want to echo that question. I'm very concerned if you would change the definition for a trial and have it be different from the published classification. I mean, if the point of the clinical trial is to influence ultimately clinical decision-making or management, and you're using a definition that's apart from the ones that people are using in their clinical practice, one adds another level of uncertainty about the meaning of the results. And you also introduced the question that we've already heard multiple times: which classification are we using anyway for any given study or thing? So, and I agree, I hate acute tubular injury as a component, but I think that should be a system-wide adjustment if other people agree.
I agree, but I think that takes us back to the discussion on what the classification is for, because I think if you're using it for diagnosis, we accept a certain amount of uncertainty. Brian was telling us yesterday that he's happy to have suspicious or uncertain things, where if you're enrolling a clinical trial, it's a different purpose, really.
I'll join in a comment and support, so to speak. And Candice, you know this. As we embarked, in fact, on an observational study, we sought to standardize diagnostics with relation to the outcome of Abmr, using retrospective data initially in that context. And this was a big effort to make sure that the diagnoses are harmonized this way. So I'd echo the concerns with regards to the challenge, and I would say that by the time we were able to standardize the automation of the process and make sure it's vetted on by multiple people, there arrives a new classification that requires some modification. So again, all of this to be said, the process is dynamic, and I think all of us acknowledge it, but at the same time, once clinical trials take years to pursue, inevitably, I agree that we need to standardize definitions, potentially collect more data, inform ourselves of how whether used as an outcome or as a potential impetus for treatment, so as a diagnosis warranting treatment, and evaluate the therapy moving forward. And all of this to say, I realize this is by no means new, or that this will not surprise anybody by any means. These are the challenges that we struggle with. But I really want to emphasize the point that you made with regards to the context of clinical trials, so that the data, the tissue is somehow, while securing patient confidentiality, is made available for us to reevaluate this, because eventually from a study design perspective, this is the best data that we could retrieve, and as detailed as we could be as we embark on clinical trials with regards to baseline risk factors, whether it's molecular compatibility and longitudinally collecting information, this is likely going to be the way for us to proceed forward. And again, we must be consistent with our definition, otherwise we are unable to accumulate sufficient sample sizes to inform care moving forward. Thank you.
AMR as endpoint, shouldn't it be the starting point in a trial for AMR? And is the endpoint the change in function and lesions?
That's a great idea.
So why is AMR an endpoint? AMR is the enrollment criterion, and then you treat for AMR, and then you monitor whether the pathology changed.
Yeah, but then so that's the question, what would you use? Bound lesion scores?
Yeah, and function.
You could perfectly consider a trial that you want to prevent AMR as the endpoint and even accept that there would be a little bit higher TCMR, exactly as we have seen.
Okay, yeah. That's also possible, but so it can be both. Like the entry point and the endpoint.
Hi, this is Amanda Klein from Transplant Therapies Consortium. Can you elaborate or talk about if there was any feedback from EMA regarding if this was a question?
Sorry, what did you get that?
Oh, as part of the qualification advice, was this question posed to EMA at all about updating?
Yes, I mean, Martin can comment better because
Okay.
we interacted directly with them. They gave an advice which we included in the paper, but I'll let you comment.
So their advice was actually that if you want to have standardization of the definition of AMR as inclusion or as endpoint for clinical trials, that this needs to be backed up by the community. So they gave it back to us. And if we don't come to, let's say, an agreement, which likely will be quite difficult, but still, the AME and the FDA, they get the clinical trial designs from the companies, and they currently are deciding on what are the criteria for inclusion of endpoints. And at the moment, this is wobbly, as you have seen. Everybody's using different ones, and so it's then the task of the people submitting the trial design to convince the health authorities that this is a good trial design. And of course, then this is sometimes backed by the reviewers of the trial design, but sometimes not. And so I've been in committees with the EMEA where we have actually refuted the proposal by companies that the endpoints that were proposed were not good. And in other circumstances, they just followed band classification but eliminated most of the things that are not universally acceptable. ATI, the molecular microscope that is there, is not proposed by those companies as definition. And this is also possible, so we can proceed without having consensus in this room. The companies will find their own way to move forward. But the problem with that, and that's actually the reason why we started the ESOT interaction, this is good for companies involved in the field, but it shies away companies that don't know all the details, and they get extremely anxious with all the, and so that is why we try to come up with some kind of easier definition. Yeah, but perhaps, I don't know if that's an answer.
The easier definition could not be like BAMF automatization, BAMF diagnosis that is recognized, and you want.
No, I think that's right. You know, how do we interpret the lessons from the past? You know, why does all those clinical trials failed? And you know, Mark, you look at me, but there is the eculizumab story, which to me is really relevant. So how do we analyze the failure of potentially really relevant drugs which never made it? First of all, problem of enrollment, problem of definition and application of the BAMF rules. We have 2 ongoing randomized trials in many centers. We collect the BAMF diagnosis and the BAMF lesions, and we apply the BAMF automation system, and we found like huge deviation from the application of the BAMF rules. So maybe that kind of system could be a gatekeeper in the clinical trials to add an additional layer of confidence with the fact that actually the imperfect BAMF classification at least was applied, and we kind of speak the same language. So adjudication committees will also serve. So my point is about definition, standardization, of course, and endpoint definition. We know in the eculizumab trials the problem was more than the evaluation criteria were not the right ones, and there was probably a change in the clinical expression of rejection. So yes, I think the standardization automation system might be a way to move forward.
And the scoring is all over the place. That's the other thing. I mean, with all due respect, that's what every study's shown is that people are not very good at scoring G and PTC, acute deletion. And you know, we had every pathologist in the Mayo Clinic system looking at them, and they got all different scores. And you know, that's the way it is. So it makes me think that I don't know anything about pathology. I'm a surgeon, right? But that doesn't make me feel too bad, really, at the end of the day. So I just think that that's really one of the issues too. But you have to have for the FDA, there has to be some sort of definition, and you have to adhere to it, and you have to just live or die with that one, and hopefully that the drug is good enough that it probably takes care of a lot of the wiggle and the definition. Very good.
Now, what I was going to say is the problem with these trials really completely with BAMF, or is it how, I mean, there's certainly the BAMF definition, but we talked about yesterday, you can have the same biopsy, but different courses of the disease. You can enroll patients who are really far gone, and then patients who are very early in their course in surveillance, for example. And their clinical course is going to be different if you have an endpoint like a change in GFR or graft loss, for example. So I think that we have to agree on BAMF, and the classification is one thing, but the scores are another, because you can at least, if you get, now put it reproducibility and all of that aside, if you at least have the BAMF scores and incorporate those into the classification, you can deconstruct the whether it's 13 or 15 later. So I think we keep it the way it is in BAMF, and then maybe have other things. Early and late should be different, de novo and pre-sensitized different as a start. That, I don't know what others think about that.
Thanks. My question grew out of your last question, Candice, and then also what Alex was saying. We're all talking about standardization. And I was wondering, does a global template exist with controlled terminology that gives all of the granular detail that we give in a renal biopsy report? So CDESC terminology or warning C, these types of things that are used in clinical trial data sets. Does that exist? And if not, could we develop one in the BAMF and the Renal Pathology Society so that there's something that we can all have as a standard data set for collection?
It was discussed in Pittsburgh, you're right, about a common template that we would use. How do we report some degree of harmonization in terms of data entry and what we report in diagnosis? It was also, with Mark as we went through all the meetings, feedbacks, and one of the feedback was clearly what you said. And Carrie is right, it's part of the equation. We also need to have the good endpoints, we need to have the response to therapy related to the fact that we have an array of parameters that we talk here about them, and I think standardization, having a common template, so that should be probably one of the prospects of the BAMF report this year, is maybe that we make another step towards and define what would be an ideal template, for reporting. At least, that could serve as something that would be enhanced reproducibility. But again, Carrie, in all the trials we have, I mean that 25%, it's not only a grading, that it's the wrong application of the BAMF rules.
Absolutely.
That's
different though than what we do, right?
Yeah.
I agree, Kylie, because it's one point to have the good diagnosis, and BAMF is very good, and to define the response to treatment, that is another point, and we have to work on this. Defining the response to treatment is not using exactly the same classification. So that's another point. We have to separate prognostication, response to treatment, and diagnosis, and not mix every time anything.
And just as a continuation to what Alex was saying, so there is a working group that has worked quite diligently, right, to develop a minimal set of criteria. So the scores, additional diagnostic criteria, and so on and so forth. And there is an initial template that has been put out there, and I'm well aware that there are multiple ones that are available across different groups. So bringing all of these together, accomplishing consensus with this regard is probably a good strategy moving forward. I think the key issue, though, with regards to these diagnostic criteria, though, it becomes more challenging if there's, again, in a context where there's additional scores that are put into consideration, and if those inform diagnosis that need to be excluded to allow another diagnosis to be provided, for example, the Abmr. But as long as people come together and there's agreement about the definition that one is pursuing, it's probably doable.
Yeah, so one more aspect from my side is this restricted definition. I thought it was very important for this DSA negative antibody-mediated rejection. If we want to include clinical trials with patients included with DSA negative and have DSA negative then as an endpoint in clinical trials, then we mess up everything and we lose any credibility with the industry or so. So I think we really have to stick towards a clear definition, and this is for antibody-mediated rejection, that there is an HLA antibody, and I even would term it an HLA antibody-mediated rejection, differentiating it out from other antibody-mediated rejection like AT1 receptor or whatever.
Yeah, that's a really good point. It wasn't on my slide, but the requirement for anti-HLA DSA was also about here.
Exactly. We can do clinical trials from AT1 Amr or
Right, but this is a different story.
Rejection.
Yeah,
maybe it's the same.
but they make different treatments.
Yes. Last. Right. The last.
Just left field. I mean, Banus has identified the ABCD paradigm where it's an incomplete phenotype. So quite often you won't have all four, you won't have everything together. Yet we want all three. Is it possible to have a suspicious, like 2 out of 3, like we used to have? So 2 out of the 3 criteria to call it suspicious? Like for the DSA negatives, for the C4D negatives, because you're getting a lot of, you don't have the full house game of cards quite frequently. A lot of them are incomplete phenotypes.
So you're thinking about trying to recruit more patients to clinical trials?
Yes. As I said, a lot of them get cut out, and I know they're antibody-mediated rejection, but because they've got coexistent infiltrates in the interstitium, that automatically excludes the PTC. So all of a sudden I've lost MVI, and I can't use MVI to call them antibody-mediated rejection. So I know they've got antibody-mediated rejection. They'll have a DSA of 10,000. They'll have transient glomerulopathy, and there'll be C4D in the gloms, not in the PTCs. And then they're proteinuria, but I can't call it. So we used to have a suspicious, but we don't. Now I know there's philosophical. I know we want to be black and white, and there are times where we can't decide, and there's a partial time.
It was not the last.
I'm not an optimist, by the way. Just saying.
I don't have an answer to that.
We have the answer.
No, I'm only going to say that the problem with including suspicious is that at least in, say for example, the Martin's studies, suspicious has a different natural history / outcome than the full-blown. So it's somewhere intermediate between the no rejections and the full-blown AMRs. So the problem with including suspicious is that now you're sort of including, it may not be apples and oranges, but it may be Macintosh apples and Fiji apples.
But that can be organized by the clinical design and the question we want to.
Yeah, but then you'd have to include that many more patients.
Or to separate apples and oranges before.
Different varieties.
It's like the natural history of including all the
Exactly, it depends what we want to see, so we can separate in the study population. So thank you, we can move to the factors. Who's next?
I think I'm next.
Have you got your job with you? Have you got your
Nice introduction.
I introduced myself.
Have you got your job?
I do. Right here in my head. Right here in my head. I've memorized it. Are you?
Okay. I did, I'm old school here. I'm not Ross Manning, that's for sure. And since I was boycotting Google, here we go. And do you need to do something in addition to my doing something? Just make sure it doesn't change the font and everything else. I can start by giving my talk and saying that I'm very happy that the band meeting was banned. It's very confusing to me otherwise. I was in the 5th grade when they first had the meeting with the rest of you folks here. And I also think it's great to get back together after all these years. And I think I'm really looking forward to this. I'm also always a little confused that they keep asking me to come to this meeting because I know nothing about pathology. That's one of my claims to fame, maybe, is I don't have much of a dog in the fight. But I talk a good game. I think that Carrie and the group at Mayo knows this. So as Mark Pesovich used to say, these are my financial interests. If your name's not on the list, just see me after this talk. I always like that. I'm asked to give a talk in the role of protocol. I call them surveillance biopsies in clinical trials, and I work at that top one, but we actually do, this is in collaboration with the other two Mayo sites, a lot of the data we're going to have today. And so the idea is, do we use surveillance biopsies in clinical trials? And the answer is yes. And we're using them more and more, I think, over time. And people still don't like doing them, but they kind of forced through by the protocols. You were going to hear about the iBox scoring system, and there definitely is an aspect of that. It may have less added value to iBox than we hoped. Tolerance trials are using these, and it's more of a safety issue, I think, in this trial. People want to see if there's subclinical inflammation. You take people off immune suppression, and I think that's a good thing. The non-invasive monitoring trials have been using a lot of surveillance biopsies, TrueGraph, and cell-free DNA studies, trying to correlate that with subclinical rejection. Disease recurrent trials use them. The issues with that, in my opinion, my perspective on this is these are mostly early biopsies. I think they're sort of useful, but early biopsies don't show you as much as late biopsies. And definitely, I think the story of my talk today is going to be, it's kind of working, but we could do better, I think, than what we're doing. There are major problems using biopsies in clinical trial designs. One of the things is people don't do the biopsies. That's a big part of it. They don't do the biopsies. A lot of patients just refuse. And so if you're not doing the biopsies, then the study kind of defaults to an EGFR proteinuria approach. Not that that's not made some people some great careers out of that, but that's basically what we default to. You're actually studying very low incident events, and I think that that's a big issue in transplant. And the biopsies have, some of the findings of the biopsies have significant but small importance to impact on clinical outcome, especially when you're using clinical factors like EGFR and proteinuria. A lot of the time, the study sizes are way too small for clinical trials. And I'm sympathetic to putting more people in with actually suspicious Abmr than having nobody in the trial at all. And therefore, I think you might be able to do, you may learn something from the trial. You may not get it FDA approved, but I think you'd learn something by doing that. So again, I'm a surgeon, so I have very kind of wishy-washy opinions on most stuff. So the early thing, I think, is the early biopsies. Early data points show a lot less injury, and therefore I think that are hard to really have an impact on long-term outcome. And that's the thing, is the early changes really may not impact late graft survival. And that's the T-cell mediated rejection these days, right? So we saw a drug fail because of high incidence of T-cell mediated rejection when it's probably a fine drug, let's be real. And getting back to what we've all talked about, but I think it's really important to say that for clinical trials, the reading of the biopsy is variable and inaccurate, and it kills the trial. So the current approach, you know this, that's poor reproducibility. The bad thing is we need to scan all of these slides. I got an email today that they wanted slides sent to them. And I said, well, we scan the slides, we can send you to scan slides. And they act like that I was from Mars, that this was something really new to them. And, but you can go back and you can ask, you know, what did Brian Mankiewicz call CG? Well, he had to circle it and say that was what he called CG. So it's very nice to go back and audit it. It's the discontinuous scoring system of BAMF has some major issues in clinical trials in that it decreases power. And that's a problem in clinical trials because you're fighting with low enrollment and low numbers of patients. There's multiple ways of scoring if you hadn't noticed. And so I think that this, the nice thing about having a scan slide, you can go back. And the BAF system was really never correlated with outcomes, and I have always tried to correlate with outcomes, but I know that's sort of tyrannical also, because if everything has to get to be a C statistic of 0.8 or have a p-value that is multivariate analysis, you're probably going to lose a lot of data, especially in the early phases of understanding a disease process. So, spine for everyday clinical management, we covered this. So, variability in scoring hurts clinical trials. We've got a bunch of these, but I think this is the best one that we ever put together. Looked at the scoring of 67 biopsies that had Abmr. And if you look, this is what I was saying here at the top, glomerulized and peritubular capillaritis has very high variability between pathologists. And they do better with CG, because I think duplication, most people can understand duplication, right? Even I can probably understand that. But the issue is that the variability is a big problem in clinical trial numbers. So it can really significantly affect the outcome of a clinical trial. So we did this thing where we had one pathologist read it, then we had this 2/3 majority, and then this theoretical study where everybody agreed it had 100% correlation. And if you look at the p-value of 0.05 over here, you don't need very many if you know exactly what the outcome is. I assumed a real histologic response rate of 50%, which is, that would be like a miracle drug for Abmr, right? Let's be real. Where 50% of the patients got better on the drug, and 100% of the other ones didn't. And if you look at that, you needed about 70 patients with the majority rules, and about 105 or so patients if you were taking, at the worst case scenario, with the pathologist who was the expert central pathologist they sent it to. So you had to have a 50% bigger clinical trial just to take into account variability in scoring. And that's a lot of money. And that actually is a problem I think that we should always consider. And I hope that someday we're not going to have to do this, because it's going to be a different way of reading biopsies. So, but it's really a problem that the possibility the drug is actually working, that it actually has a 50% reduction of Abmr. And you don't know that by the p-value. So I guess the other thing is really what we're looking at, when we're asking these questions about using histology in clinical trials, really asking ourselves, are we going to really improve Cox models when we do any of these? And of course, all of this is based on having accurate outcomes data. And if the outcome is, let's say, graft loss, we don't have outcomes data, very many long-term outcomes data. We have to have a very large data set because the events are rare. And again, getting back to that accurate reading of the biopsy is going to affect that. And I think all of these right now are severely lacking in clinical trials. And using surveillance biopsies in this is just going to be adding to the problem. Just to say, there are times when we have shown that surveillance biopsies have actually improved the predictive value of clinical trials. And you probably know the iBox system. You ever heard of that? I think it's been presented a couple of times in the last couple of years. And but the idea is that it's basically EGFR, proteinuria, and DSA plus minus histology. So it's not the most sophisticated system in the world. We've been doing this probably since before I was born. But if you add histology in our calculations, it does improve the C statistics of 0.84 to 0.9. But more importantly, it improves stratification, which is really what I think at the end of the day a clinical trial is trying to show, is which of these people are actually going to lose their biopsy. It's not, and that's that I think that we can look at that, and it was, for us, it was G scores and CI scores, and I think for the iBox it was PTC, and but basically it's Abmr and fibrosis is what we were able to see. And fibrosis is in all of these models, but it's actually has very high variability in scoring also. So our group has actually focused on more accurate scoring. We're looking at more at continuous scoring, and I think that the future we're looking at is computers versus human scoring. And I'll just take a couple minutes to go over what we've been doing in the last few years. You may have seen some of these before. We've used morphometry extensively, really working with one of our collaborators at Mayo, Andrew Rule, and we did a large risk study. We've looked at a lot of 5-year biopsies that had subsequent graft loss, and we compared BAMF histology to our morphometry with this. And these are the way that we do morphometry. We publish this. And the nice thing about this, of course, these are scan slides. We've gone back and circled everything, so you can go back and scan and look and see if you agree. What is amazing about this is the reclassification that we've seen. You do see that, 0 is 0, and the MN3s really do have more mesangial expansion, for example. AH is the same, fibrosis is the same. It's just that the cutoffs aren't right a lot of times. And there's a lot of overlap. And I think that, back in the day we used to talk about, the biopsy, there was a lot of variability in sampling. But I don't think really that's the major variable in biopsies. I think it's really scoring is what we're looking at. And, I don't think that this is perfect, but it does tell you that there's a lot of biopsies that have many different ways of being read, I guess is the best way of saying it. And every time that we looked at morphometry, it was a continuous scoring system, and it correlated with outcome better than the Banff scores. And over time, they started to correlate better with long-term outcomes. And our emphasis, probably just because my interest, is in long-term outcomes. And so we usually start with our 5-year biopsies when I'm talking about long-term outcomes. And if you look at all of these improve the Banff scores when we use we're better than Banff using morphology. So both Banff and morphometry identified lesions associated with graft loss, but the manual morphometry models predicted graft loss better than Banff, and also, I think importantly, in biopsies without CG. CG is one of these lesions that when you have it, it's one of these lesions that graft is going to be lost to that process. But our group is interested in other things besides just rejection and CG. And I think as you get out further, if you really want to improve long-term graft survival, there are a lot of non-immunologic events that I think that we should be studying and also be putting into clinical trials. And I think that Banff identified more features that are predictive of graft loss that you guys have told us are important to look at because it's part of the Banff schema, but we just never look at them or score them right, at least our group doesn't. And so I think that Banff models, that the computer system morphometry are predict better outcomes than Banff. They're more producible, they're auditable, and I think they could be developed into biomarkers. And just to say, 2 cents is that we study 5-year biopsies because that's sort of the beginning. I think we're starting to see more chronic lesions, and that's when you're starting to see truly a separation and seeing a lot more graft failures. It's great. We're trying to nail down a endpoint or a secondary endpoint for clinical trials using iBox, and that'll be great. But 5 years, there just aren't enough events, and many times to see the outcomes that we want to see. And so that's one. And I would also say it's not all inflammation. Here, since I have the lectern, I'll tell you this. One of the things that we did recently is we have looked using manual morphometry and also now AI at global glomerulosclerosis, ischemic glomeruli, and by morphometry, if you have more than 40% ischemic glomeruli, you can see that this is not a good thing for your kidney long term. And but this is not just inflammation, right? The inflammation is definitely in there, but it's not every single biopsy is like this. So there are other lesions, I believe, that you can see that are going to be important for us to look at. And one of the weird things that we did is we looked at MM scores in diabetics who were obese, and they actually predicted mortality more than they actually, just about as well as they predicted graft loss. And that's because I do think that a 5-year biopsy with recurrent or with diabetic nephropathy on the biopsy or mesangial expansion is really the early findings of it. That is probably indicative that diabetic has a very inflammatory milieu and is trying to kill their kidney from mesangial expansion. So the next phase I do think is going to be AI, and I think that pathology is really going to change in the next decade or so. There's going to be more talks on AI, and we are collaborating with a couple of groups doing AI. And this is just one of the first early papers, but there's a couple of papers we have that have been published or in press since then. And I think that our approach to AI, or to deep learning models, is to use morphometry as the ground truth. And because I do think we still do the majority rules to to get an idea of what's in the biopsy, but I think that it turns out to be, from our perspective, probably a bad method of training an algorithm on a system like this. And it can be audited. There's a continuous score, just like the manual morphometry, and I do think that it's going to be more accurate in the end. So I think that we're going to be having arguments here in the next 10 years about which algorithm predicts outcomes better than the Banff scores. And I think that that'll be a great day, and I want to stick around long enough to be at that conference, because I think it's a very achievable goal. I think that one of the things that we already know is that there is a lot of data in this biopsy that we are not capturing with the current way that we're reading biopsies. You guys do a great job, but I just don't think there are ways almost of expressing this. And I think one of the things is we are really losing a lot of important data from biopsies that we're doing right now, especially when we're trying to put them into binary classifications. And I'd like to see this more like what a kidney biopsy looks like, the readout, which is a numerical thing. This is what one of my techs sends me all the time when I ask him to send me data on like diabetic and their biopsies. He sends me these things. And I think the last thing is I would also put out there, as we are reviewing papers, many times we are trying to correlate some sort of histologic findings with an outcome, and it may fall out in a multivariate analysis, but I don't think that that's necessarily meaning it's not worth studying. It's just that we aren't really scoring it necessarily perfectly right now, and that I would still look at the univariate models and also subsets of patients as we go through, because there are probably a lot of histologic lesions that we are not studying that are going to be important. The acknowledgments, I've had a very great group here to help me through the years, and we continue to collaborate. And this is our group as doing our team bonding. It's not exactly Banff, but it was a fun day anyway. Questions?
Thank you very much for this very nice talk. I'm just wondering, why do you think Banff should be able to prognose anything? It's not done for that. It's a diagnosis classification. So it's a discussion we have this morning. I'm not sure we should mix the two. I think they are two very different things. They need different things. So I don't think we should go for one single thing doing both diagnosis and prognostication. For instance, I'm not sure that the Excel sheet is helping us to know what the patient has a disease.
Well, my thing about always has been, we definitely use the Banff scores, but I think that, and I think the Banff, the classifications that, the columns that things go in, those do fit a certain profile, and we tend to use those. Mesangial expansion, CG, inflammation, we still study those things. I think that one reason I have a problem is the categories that we throw them into, the binning of the categories. When it turns out that by the eye estimation, those numbers aren't very good. They aren't. That's just what we found over and over again. And maybe it's just our people, but that's the problem I think you have with Banff. And you're taking this continuous data, and a statistician will tell you, don't do that. Don't try to dichotomize the data. Or at least look and see where the inflection points are. And I think that that's, again, I'm trying to be provocative more than overly critical of everyone. But I think the 0, 1, 2, 3, and it's just a gut. It's sort of human nature. You have nothing, you have a lot, and then you have maybe yes, no, and then you have it's really here, but it's not a lot. And that's 0, 1, 2, 3. And that's the reason we change the numbers and change the thresholds and all this kind of stuff. And I do think that it's not that we're trying to get not to have this meeting ever again, kind of a thing, and it's going to be reduced to some sort of formula. But I do think that there's a lot of data on biopsies you lose when you use those kind of systems, and the variability is something we have to, if we really want to improve patient outcomes, I think we're going to have to somehow come to grips regarding that. And I think morphometry helps a little bit with us with the variability. It's not perfect. People can just do a little bit more in there. But I think that you can look at a biopsy and say, okay, you've probably circled all the fibrosis. And then you push the button and, you know, we have people guess. And people guess 15%, some people guess 50%, you know, and it's like, and every time I wanted to do morphometry, everybody hates the idea that we would do it. But I think it's an interim step toward training models to do it.
The last sentence you said you circle all the areas of fibrosis and then you get it. Does that mean that you have a human using their eyes and circling the fibrosis?
Yeah.
And coming up with a number? So that also has bias of what we consider fibrosis in there. So that is not the state-of-the-art system. There are trained algorithms, and Brad Harris has published on that with image analysis and having stains for different areas. So clearly there are different ways. But circling all the areas of fibrosis is a clunky way that has its own errors and biases in it, in my experience and opinion.
But then the question, I guess, is then, there is no gold standard, right? You have to be some sort of godlike individual, because you can have two people who can look at the same biopsy and circle it.
There is a gold standard. There's a gold standard for fibrosis with serious red staining assessed by morphometry under controlled conditions with polarized light. That would be a fairly good gold standard.
Well, again, the study, I'm not saying that morphometry has its limitations or doesn't have its limitations. It does, more thing is how we're going to do this in a continuous scoring system way, right? And I'm not advocating morphometry for any clinical use at all, because I think that there can be great variability in some of the scoring systems we use. And I think that it would be nice, we're trying to include the pathologist more and more in these kind of things. It's just not the easiest thing for us to do. I know Brad Ferris very well and what he's done. I just wonder why we haven't moved more in this direction as a field, especially for clinical trials where reproducibility is an issue, where scanning of slides and doing some sort of morphometric analysis might help us interpret the data better. That's kind of where I'm kind of suggesting as we do as a group. I mean, I'm in an ad board, but there's no way they're going to do that, right? Yes?
It's quite easy just to, you can get your pathologists to eyeball the percentage, so you can look at, say, 12% going to 20% that stays within the same category, but you can have a series of numbers. And if you're looking at 2 protocol biopsies or follow-up biopsies, and that is your primary endpoint, you're looking at a change or a delta, you could do that, yes. You could overcome those quite simply just by getting a more granular number, or a computer AI, or morphometry. Or you could just choose another endpoint, so like GFR. So that's what we would do, and you have lots of GFR measures. You don't have the problem of, no, I don't want to have a biopsy, yes I do. And that's predictive of end stage. So you can use, it depends on how you want to use your biopsy, whether you truly want to include that as a repeat process, and that's going to be one of your major endpoints. Then you have to work out, well, what am I going to use? Is it going to be a delta CG, or is it going to be one of the acute lesions? And the acute lesions aren't all that reliable. Did you have any thoughts on if you do a repeat biopsy, or does anyone, what you would use?
I would advocate, well, I think that a lot of these questions have to be done in the context of a clinical trial at some point. Really a studied way of looking at, maybe we do have something that improves inflammation at Abmr. Then you have to score inflammation, right? Score the delta inflammation. And I think that one of the things I would suggest is to do some sort of staining so that you could count the pixels or something of that nature so that you could see a delta inflammation. Whether that would correlate with better outcome, who knows? I think there are instances of like the Eculizumab study where we prevented acute, but chronic still happened. And I think that, and that's a study that you get a fairly quick readout because it's antibody, right? But for studies which are going to be long-term studies, I think that the iBox scoring system, all those other systems would have a higher C statistic if the biopsies were read in a continuous scoring system. And I think that, therefore, I would not throw out histology as an important aspect of clinical trials. It's just not being done in a way that we can really get all the data out of the biopsy.
Clemens Wuder from Charité Berlin. The topic is lessons learned from the rejection phenotype in recent clinical trials.
Yeah, so thank you very much for inviting me. And again, I thought about a long time about how to present and how to present it, because it was kind of a very, so not really intuitive task Martin gave me. And we heard a lot that clinical trials are crucial to advance the field. And of course, the question is then again, what can we learn from this? Here are my disclosures, and I would continue with Mark that everyone can contact me if he's missing on the list. And basically, yes, and I start here with the Symphony Trial, because this was the most influential trial over the last decades. It really has set a new standard of care. And what lessons can we learn from these curves? And you see basically the lower curve is the TAC, low-dose TAC arm, and you see basically most rejections are in the first month. It's all early rejection, later on just a few more rejections. The rejections were read by the BANF 93 report, or basically the BANF, we heard the first BANF meeting ever. So the good thing is that it's the BANF classification based on the Symphony Trial. The bad issue is that when we publish next year our report, then in 16 years, so in 2039, a study will cite our outcomes. So it takes a long time. This is not really surprising, because it takes time to do the trial, to publish first the BANF meeting. It takes time that someone has to read it, then to study, to plan the trial, get the money, and then do the trial, and then finally get it published. But at the end of the day, TAC-MPA became steroids. We have 50% rejection rates, mainly TCMR, again, and this 16-year delay, which is we have to accept. Then another trial from Germany, from Symphony to Harmony, again a trial on steroid-free treatment, 3 arms, all TAC-MMF, 600 patients, so large clinical trial, 8 days of steroids with or without ATG, or just continuous steroids. And you see here rejection rates and graft survival are not distinguishable, and so it's kind of nice to get the lessons. Again, the study was started in 2008. They used the BANF-05 classification and published in 2016, so it took 11 years after the BANF classification published. The endpoint, again, 11% rejection, 80% T-cell mediated rejection, and 80% T-cell mediated rejection BANF class 1, because there's only very few patients with BANF greater than BANF. Antibody-mediated rejection, according to BANF-05, was 1, so 8 patients out of 600 had an antibody-mediated rejection. Yeah, should we talk about this as a big problem? And then it was steroid resistant, 2%, again, mainly in the first month, and excellent patient and graft survival. So can you get better? That's the question. Low rejection rates, excellent outcomes, steroid-free. Again, focusing now on other endpoints, which may be much more important for our patients. For example, diabetes, have insulin or no insulin, and have dietary restrictions. And then maybe the rejection is kind of stepping back as an important endpoint. The problem is it's only 1 year follow-up and low-risk patients. So a study with 2-year follow-up, comparing TAC versus TAC. So you see basically both arms are the same tacrolimus, and you see again after 2 years there were 18% rejection. Excellent outcomes after 2 years, very few death and graft failure. So again, when looking also for comparability for the 1-year data, after 1 year there was 14% rejection, again all BANF-1 rejections, antibody-mediated rejection, I have to admit, and this is, I published this study we didn't even report. I don't know why, but obviously it was rare. Mainly in the first month, again, the rejections. After month 12, there were from month 12 to month 24, there was 3.5% rejection, again, and excellent outcomes. And when you look now in the more recent literature, Transform study or other huge or large trials published in the last years with TAC-MPA, you always have rejection rates of 10 or even lower percentage, and this is kind of has become the standard of care. So what about maintenance patients? And actually rejection rates in maintenance patients from clinical trials has been rarely reported. They're basically, I think, only 2 or 3 or 4 trials. One of this is again a comparison of TAC versus TAC. So LCP, TAC versus Prograf. And the rejection rate, you see 1.2% in this maintenance population. So it's really, basically, it's extremely low in those maintenance patients. Again, this is a selected population, it's a trial population, and the trial population is of course maybe more adherent and more, has a closer follow-up. Another trial for maintenance patients was a recently published BELA study, BELA conversion study, and just focus on the CNI arm, and you see in the CNI arm, over a 2-year period of time, 2.7% rejection, so 1.5 or 1.3% per year. Again, all BANF1 rejection, and there was one BANF2 rejection. In contrast, however, BELA patients had 9% rejections after conversion, and you can see a lot of BANF2 rejection and also even BANF3 rejections. Human rejection, 2%, low in both arms. So in summary, CNI plus MPA, 1.5 to 2% rejection per year, 2% Abmr per year. BELA, more rejections, however, less DSA and better renal function. And then coming back to the BELA studies for registration. And you see here now, BELA has more rejections. Again, as BELA is not as potent, obviously, as TAC or CNI. This was a cyclosporine treatment arm, and you see even in the cyclosporine arm there was low rejection rates, and the BELA patients had really significantly and relevantly more rejections and more severe rejections. Again, this signal in BANF2 rejections. So again, with cyclosporine was excellent rejection prophylaxis, BELA more and more severe. And here the rejection over time, and as you can easily recognize, all rejections, or mostly rejections, occurred in the first 3 months again. In the cyclosporine arm, it's kind of ticking up a little bit over time, but BELA, you see the red line, for example, basically after month 3, its game is over and there's no rejections anymore. Again, after year 1, there were few rejections, and for rejections you have to pay a price, so that's for sure, so the GFR declines. But for cyclosporine people, even if you have cyclosporine and had no rejection, you have worse GFR compared to BELA with rejection. And of course the hypothesis is maybe then if the cyclosporine associated nephron loss is maybe more important than a rejection. So do we need another focus in our transplant field? And another interesting thing, and this has been also reported in many other issues, that T-cell mediated rejection is a risk factor for DSA. And this is true here for the BELA study, but also and more for cyclosporine. But you see that even patients without acute rejection in the cyclosporine arm had at least twice as much donor-specific antibodies compared to the BELA patients. So again, very simple, the rejection is associated and maybe should be avoided for this reason. The other hypothesis at the time was, of course, acute rejection leads to chronic rejection or chronic allograft nephropathy, as it was called at this time. And just remember, in the patients in the benefit study, they had the most rejection. That's twice as much rejection as compared to cyclosporine, and they have half the number of chronic injury. We also had the hypothesis that patients at extended criteria donor are especially vulnerable to CNIs, and that this is maybe the population who would benefit most from a CNI treatment. But again, our hypothesis was wrong. Obviously, they have the same chronic damage after in this protocol biopsies after 1 year. So again, anyway, no obvious association of acute rejection with chronic injury. We have to summarize. And then, of course, the idea is chronic injury or rejection leads to inferior outcomes, and we saw again in the benefit in the old study for this extended criteria donors, they had more rejections, but a similar amount of chronic injury and similar outcomes after 8 years, so really a long-term study. But again, there was no obvious effect on acute rejection or on cyclosporine treatment in this vulnerable group on long-term outcome. They all had the same, so the kidneys did what they did, or maybe we could not influence it. Contrary, the study where we had the highest rejection rate, they had much better outcomes compared to the CNI arm. And really questioning our hypothesis that BANF2 rejections, and this were mostly BANF2 rejections, having detrimental outcomes, or otherwise, so we have a 43% risk reduction in death and graft loss despite more and more severe rejections, so twice as high rejection rates and much more BANF2 rejections. But you have better renal function, better histology, and less DSA. Again, questioning, or the hypothesis then is, if you can treat rejection, maybe it's not as damaging as we thought. And obviously, or seems to be, that the BELA patients, the rejection was treated successfully. Otherwise, you could not explain those results. And even were treated with a minimal amount of nephron loss. So in summary, part 1, I hope to have convinced you that rejection is not important anymore because it's less frequent. It occurs only in the first 3 months. After that, we don't have to worry about it. That it's really low in maintenance patient, 1 or 2% per year. That it's almost all exclusively BANF1 rejection. That ABMR and BANF2 rejection don't play a role in clinical trials. And also steroid resistance is rare, so treatment seems to be successful, and the results from BELA studies further challenge the importance of acute rejection. So is it really a relevant clinical problem anymore? Yeah. Again, we did a study similar to Mark Stiegel, and Mark did a study in the Mayo Clinic in 2009 looking why do we lose kidneys? And I think this is really a fundamental question. And we looked into this and really came to similar conclusions that medical events were the most frequent reasons for graft loss. Pyelonephritis, infections, cardiorenal or whatever. The second frequent was T-cell mediated rejections, followed by ABMR, and then CNI toxicity, transplant quality, recurrence, polyoma, perioperative. But when you put it together, rejection and ABMR and TCMR were involved in 65% of graft losses. So rejections still play an important role in reducing the nephron number in a very vulnerable kidney. TCMR was associated many times as a secondary cause, somewhat in conjunction with poor donor quality, as Mark Haas pointed out, or in conjunction as a T-cell mediated rejection was followed then maybe by ABMR, and then we lost the kidney, while ABMR in 30% of graft losses involved was the most important primary reason for graft loss. So it seems to be important as well. But we also looked at another issue is non-adherence, and 20% or almost 20% of cases were non-adherence, and half of them had rejection. Pointing towards the issue is that rejection is not always an immunological phenomenon. We have to look at psychosocial issues as well. And if patients don't take the drug, as someone said today, we of course have no effect. So I didn't talk about ABMR so far, or only mentioning that it's not really important or only rarely existing in these trials, of course, and these trials were always with patients with low immunologic risk. And only short period of follow-up. Again, when we have talked about ABMR, we have to talk about the different classifications. This was pointed out several times, and Martin has shown that even this classification has some effect on the different, or this classification has changed, and so the outcome or it may be wrong to have patients according to BAN 401 and compared to BAN 13, and that the outcome maybe would then be different if we would have used the other classification. Looking at this antibody-mediated rejection problem, I will show you another big study we did in Germany, the ZEUS trial. The secondary endpoint was rejection, and you see that at randomization there was kind of a small difference, and then the everolimus patients, they were randomized to everolimus or stay on cyclosporin. The study started in 2005, BANF was 97, and the paper was published 2011, so this is really the time frames you always have to look at. At the end of the 1 year, there was no difference in rejection rate, but of course, everolimus patients had more rejections. But it was again mainly band 1 rejections. We also had a long-term follow-up published in AJT at 5 years. Again, with everolimus, you had more rejections. You had more band 1 rejections, but only a very few rejections, but ABMR rejections. Of course, the issue now comes up that nobody did regular DSA screening. And if you don't do DSA screening, you cannot detect it, obviously. Only 53 out of the 300 patients had a DSA screening. Again, overall survival was still excellent. So the ABMR and the DSA obviously maybe may take some more time. And Lutz Liefeld from our group put together our patients together with other patients who were converted to everolimus and published then this paper in AJT. And you nicely can see that everolimus is not as potent, and then after 1, 2, 3 years, the DSA pop up. The longer the patients are on the drug, the more DSA they get. And then with a delay of 2 or 3 years, then the ABMR go up. And I would say, and Lutz is now finishing the next paper on the long-term follow-up, now 10 years, and then after another 2 or 3 years, then the graft losses turn up. So this is the natural history, but you have to have then 10-year follow-up to show this. So the issue is, of course, is that DSA seems to be an early biomarker for ABMR, but you need long follow-up. And we're talking about 5 to 10 years. Looking at another trial with everolimus, a large trial, almost 1,000 patients. All again, we started with CNI and MPA and steroids and Simulec, and then 700 patients were randomized to continue on CNI or everolimus. The primary endpoint was change in GFR, but we are looking now at the rejection rates, and you see that everolimus again had higher rejection rates, and TAC patients only had 3.5% rejection rate, so really negligible. Antibody-mediated rejection was a clear signal here in a larger trial confirming our data, but I want now to look at the DSA data, because for diagnosis of antibody-mediated rejection we need DSA. They had a central lab, and they had a fixed threshold of 500 MFI values. At month 3, only 60% of those 700 patients had DSA available, and at month 24, only 35 to 46% had DSA available. Yeah? Of course, if you don't have DSA available, you cannot really make good conclusions. And then class 1 at month 24, looking for de novo DSA, there was 8.9% versus 3%, so confirming our data. But the main issue I want to point out is that now class 2 antibodies, which are the main or the most frequent antibodies occurring after kidney transplantation, were not different, but were only measured in 36 or 45 patients. And why is it? Why are not those don't have DSA? Because the donor DQ typing was not done. Of course, to detect a DQ DSA, the most frequent one, you need a donor DQ type. And if you don't have a DQ type of the donor, yeah, you don't have a rejection, or you don't have a DSA, and then you also may not look for antibody-mediated rejection. The other issue is the fixed MFI threshold, and Anita Tambur was here, I think. We can discuss this issue because this is another issue. I just recently reviewed a paper. They used this threshold of 500 and found 22% DSA, de novo DSA with beta-sept. I said, oh, this cannot be true. You cannot get antibodies with beta-sept. And I asked them to revise the threshold. And then they did this with repeated measurement above 1,000, and then the de novo DSA was 0%. 0. Yeah? And so you really come to kind of interesting conclusions when you think about it. And I think this is really crucial for our field here as well. And of course you need to have, as a starting point, donor DQ available. And this was not done, or was not done. And then finally I come to a large trial for a rare disease, Abmr. And this is what Mark Stiegel just pointed out to us. This study, this was, again, the study start was 2011, and they used the most recent band classification. In 2007, the problem of the study was late publishing, but maybe Mark can tell more about this. They had 104 patients, so really a large trial, and they had an endpoint as treatment failure with antibody-mediated rejection on central pathology, graft loss, death, or discontinuation. Graft loss, death, and discontinuation is easy to count, but then AMR 2 or 3 obviously was more difficult to count, and Mark pointed out to this. And again, the central assessment was 5 patients versus 7, not significant. When you look at the local people, they had 7 patients to 15 patients. So it was borderline significance, but there was a poor kappa value, as Mark pointed out, of the grading, especially the grading was a problem. Then they did a central reassessment, and they really tried to figure out, and I think this is the most interesting part of this paper, or one of the interesting part of the paper, why does this happen? And then they provided the pathologists with more information on the patients and the clinical history, and then the kappa value went up. And what was the relevant clinical information? The date of biopsy, reason for biopsy, renal disease, diagnosis, and so on and so on. So really, a pathologist also needs, this is we discussed yesterday, needs clinical information, otherwise he cannot be good. But then the question is, what about central pathology? How you can do central pathology, because can you provide those clinical information? So I come to the same conclusions as Mark did. There's obvious problems of reproducibility. The correct pathological diagnosis requires clinical information. But the main point I also want to make here is, this is a trial where the company has to decide to spend $100 million or whatever to go or not to go. Yeah? And when you don't make the decision in the board, when they see those data, they don't make the decision. They say, this is, if we get the endpoint not reproducible, we don't make any decisions to go into the field. Yeah, this is for sure, and I think this is a really important point in this context. So we have to have good and precise, clear-cut definitions, as Mark pointed out, and the others as well. And here again, this is Kerry Shinstock's data. Again, this local diagnoses are difficult, and we have problems and struggling with this. Maybe too many footnotes, but again, we need to have this restricted Abmr definitions, and I think the way of the ESOT communication with Candice is a really important step forward to this, that we have some kind of a restricted definition which we can use for large clinical trials. And there is one large clinical trial at least ongoing, and this is the CLATA study. And they use, as we discussed, they want to include chronic active Abmr patients according to band 15, so we cannot really complain about this. They have a central lab, and this is painful, and also we wanted to send in digital slides, and they said, no, it's not possible, okay. But this, and they had a DSA central review, because the other issue is to have really reliable classification of DSA, and they also don't trust the local labs. But they don't do it again, but they have only the central review of the data. But they don't trust in Abmr as an endpoint. They use GFR as an endpoint, which is somewhat easier to measure. and is really more reproducible, I guess. So again, important information from this is what do we learn? Yes, they use a restricted ABMR definition, and they don't use it as an endpoint, but use graph function. So in summary, part 2 on ABMR diagnosis, obviously we discussed this, is difficult, but also DSA determinations are difficult. And de novo DSA, of course, is a surrogate endpoint which precedes ABMR. But for large clinical trials and for decisions in pharma companies, we need somewhat easy to follow, and I would say not necessarily simple, but easy to follow and clear-cut definitions for inclusion and endpoints, are basically the same conclusion as Candice. And for this, I would like to thank you.
Dear Clemens, you are very provocative today. We don't need histology, DSA, 100% to 0%, and ABMR doesn't matter. Please, do you have questions?
I just want to make a few comments. First, I'll comment on that Acknowledgments trial, which is interesting for so many reasons, including the histology part of it. But one issue with that was that they didn't have a standardized definition of positive cross-match, and there hasn't been a standardized definition of that. And Ruth maybe can speak more to this, but I mean, that's one issue we really need to come to terms with. It's not all, you know, it's a pathologist suck or whatever. I think that, you know, these definitions need to be addressed for who do you enroll, you know, what is the definition of positive cross-match, not just whatever they happen to say at the local hospital. Also definitions of glomerulitis. And there's no reason that, you know, that the clinical trials need to have the same definitions as for rejection as a routine clinical practice. You know, if you're looking for something like a very early transplant glomerulopathy, you know, can you have a particular way to evaluate the slides for that? That's perfectly fine, I think. And I don't know if it means that the BAMF criteria that we're using in our regular practice need to change, but we can have different, you know, and maybe more precise definitions, especially for clinical trials purposes.
But again, I would, what Charles said, so I think this is important. We cannot have different definitions. We should stick to one definition. And again, for our trials with Georg, so we made even sure that we have the MMDX on board as well, as really to make sure, and the success of a clinical trial is the inclusion criteria, yeah, or exclusion criteria, because if you have a precise population defined, then you can get a precise answer. If the population is wide over, then of course you have a lot of noise, and then you need much more patients, much more costly and so on, and then you have a higher chance of failure. And for this, I think it's really important to have, and this Mark pointed out in the power calculation and so on, if the endpoint has a high variability, the number of patients goes up and $1 million go up. Yeah? So it's really not trivial, and we really should have a somewhat easy to follow definition. And I think this is really crucial to have a good population defined, and then you can get good outcomes. But if you have, again, if you do plasmapheresis study and have patients without antibodies, you will not have success, yeah? Or at least I would be surprised if I see a success.
Yeah, in a way, BAMF has almost been a victim of its own success. Like, everyone relies on it so much that we don't realize the intrinsic problems that maybe they aren't conducive to being part of a clinical trial. So, but I think there's no, if it's working in our regular everyday practice, that's fine. There's no reason we can't have a separate set of criteria for clinical trials.
Yeah, but again, you could say even, okay, the BAMF 91 did work because the Symphony study worked, and we rely on this, so why do we need any further development, yeah? But we have the feeling that it may be needed, yeah. And I think to try to be more precise is a good goal.
Yeah, so Clemens, two points about the two clinical trials. Two of the clinical trials you mentioned. The first one is eculizumab. The other one is the belatacept trials. So first for the eculizumab, and we published that in JSN. We had the chance, not with Alexion because they didn't want to give. Okay. I thought Kim was like, just a second. Hold on. You're wrong. So first of all, we reassessed, and Alexion did not provide the data, but we were able to ask the PI of every single center in the eculizumab trial to send the data, send the sera, send the biopsies. And two very important information, we published that in JSN. It was the reassessment of the trial with some of the tools that we are commonly using in research. First of all, we realized that in the eculizumab trial, 30% of patients who had non-complement binding antibodies receive as a sole agent to prevent AMR complement inhibitors. So it's the pathogenesis-based assessment of clinical trial in the inclusion criteria, which was the first thing missing. Point number two is that when we run gene expression assessment on those biopsy, we could actually capture a dimension of different signals related to the Abmr process, NK burden, macrophages burden, endothelial associated activation transcripts, which shows that actually you had much higher signals in the non-eculizumab treated patients. So, and if you exclude the ones with non-complement binding antibodies, the study would have made the primary endpoint. So I think that's a very important lesson to make progress. And the second, of course, is the belatacept. And I really like what you showed, because I completely agree with you. We have seen those nasty rejection with V and INT. We treated them as steroid dose, everything disappeared, and finally your hard endpoint is there. I mean, it's just it made it, and it echoes the Novartis CFZ, failure of the CFZ. The study was stopped because of BPAR, the same kind of thing that we would expect to have seen with the belatacept patients. And unfortunately, now that they have the data at 1 year, what they see is that despite the higher incidence of early BPAR in those patients, CFZ, they have slightly better function, less fibrosis, less DSA. So BPAR was, in that time, a major failure that collapsed a potentially interesting drug and an entire development and a program. BPAR has a C statistic to predict long-term allograft loss in eyeballs, and we'll be talking about it in the qualification process, 0.62. How can we rely on BPAR as an endpoint? And for the belatacept, it's a good example. So I would like to have your thoughts.
Yeah, so I think first of all, I think the eculizumab story really points out that you have to have good inclusion criteria, and then you can make it. But on the other hand, pharma is of course difficult, and they look at money and they have different competing interests, and then there's oncology department and they say forget about this transplant nerds, they have no endpoints, no nothing, no numbers, so let's go give me $1 million, $2 million for our trial in oncology, and this is reality. And I think this is why we have to be aware of, and I think again the surrogate endpoint like iBox or so may make clinical trials more easier and more feasible, and I think this is what is needed, and again for all registration trials the BPAR was the endpoint, and we looked into this and it was BPAR and not even differentiating ABMR and TCMR. Yeah.
So we have Amanda here from TTC, and her question is, how is she struggling with FDA and EMA, and what kind of arguments, experts, and data she should bring to the agencies to say, come on, I mean, we should reconsider that. It's harming the field, and I think, Martin, you were about to speak, but you made that completely clear in the ESOT papers.
But Alex, just one point. So again, I made the convincing point that TCMR is maybe not so important, but on the other hand, we lose kidneys because of TCMR, and patients suffer, and they get diabetes, they get then steroids, and then they get CMV, and then they have other problems. So it's still a relevant clinical problem, and I think this is we also have to keep in mind.
Sure, but they keep the drug without knowing that long-term hard endpoint you could measure that, and infectious complications, patient deaths. they take just early BPAR as the thing that, and they shut down the entire program with that. So of course, we are not asking to say, okay, discount BPAR, we just do some IVOX at 1 year, and we just not follow the patients. And so the context of using is different. You can use something to fast track, but then you keep following the patients for infectious complications, cancer, deaths, and graft failure.
But Alex, we need a mindset change because over the last 50 years you looked at BPAR, and BELA even failed because of the high BPAR rates or whatever, or it was not, or the high price, I don't know, or both. But we always focus on rejection, and we forgot the patient on the other side that they get infection and die because of cardiovascular death. And have diabetes and so on. And so the mindset change from rejection to other patient-oriented endpoints, I think, is really relevant, but we have to keep in mind. But we should also not drive into the other direction and completely ignore BPAR.
So what's the pathway? What should we do? What should BANF do? What should...
What I propose is that we listen to the next 3 presentations after the break, and then after that we have some time for discussion, because you are pointing towards things that we are going to include in our presentation. Then we can discuss whether BANF needs to take that into account or not. I don't know if you have time.
Maybe to discuss at the end, because as we have seen, a little part of clinicians, a part of methodology, and a part of histology, and a part of DSA, so everything.
So bear with us for a few seconds while we set up. Peter Nickerson is joining us remotely to talk to us about the future of designing trials for rejection treatment.
Thanks very much, Candice. Can you see my slides on the screen?
Yes, we can. Thank you.
All right, thanks very much. And I'm sorry.
Can you? Sorry, one moment. Can you hear him?
Yeah.
Okay, everyone can hear you. It's good.
Great. And I'm sorry I'm not there. I actually really wanted to be at the meeting, but I'm enjoying the session so far. Some disclosures, and we'll talk about investigational drugs. I hope to talk about the causal pathways as part of building on the discussion we've been having, the impact of TCMR, Abmr, and then discuss endpoints for TCMR treatment trials and Abmr treatment trials. So I don't think this is a big step to what we've been talking about. Graft failure is really preceded by a loss of function associated with fibrosis, either the interstitium or glomeruli. Obviously, we can have recurrent disease, and what we've been talking about in this session is TCMR, either subclinical, clinical, DSA leading Abmr, subclinical or clinical, and then getting into smoldering rejections, as Bob Holden would call it, leading to fibrosis. And then the trigger for a lot of this is HLA mismatching and or the presence of preformed memory. Obviously, it's either fused injury manifesting with really graft function can be a problem, as can be older tissues that don't repair as well. And of course, what we do is give immunosuppressive drug therapy, try to control this inflammation. And that leads to associated toxicity, as Clemens was just talking about. Toxicity is related to metabolic dysfunction, infection, cancer, on a background of comorbidities that our patients have. And of course, what we do then is we tend to minimize drugs or the patients themselves minimize their drugs through non-adherence, which leads to an increased expression of TCMR or DSA. And so what we really need is novel drugs with improved efficacy and lower toxicity, not only to prevent, which I think is where the majority of the discussion has been, but also the treatment of BPAR, whether we're calling it TCMR or Abmr. Now, I want to just remind people that our own data has been showing that not all people are at the same risk for developing a DSA or Abmr or a T-cell mediated rejection in the first year. The degree of mismatch, while crude, is giving you a measure of who's at increased risk of having these events. And most recently, we've published the fact that as you go up in grade from borderline BAMF1A to greater than BAMF1A, we're seeing that the intermediate and high risk category 23% of our patients had a event, and by follow-up 5 years, we're approaching 30%. Most of these were first TCMRs. Now, in these TCMRs, we were looking at diagnosis of borderline as representing the first TCMR. The mean time to these events was 6 months. And then ABMR without first TCMR was actually relatively infrequent, actually compared to the 22 months post-transplant. 30% overall were having an event in the first follow-up. Now, I think the good thing we have to think about is what's happened since we went from cyclosporin to FDA-based immunosuppression. And this is our own data looking back at the cyclosporin time. So cyclosporin and Abmr, only 36% were having no TCMR. There was a lot of other diagnoses going on here. But when we went to FK, we saw that now the majority of patients had no TCMR, and the 30% was spread out over the various grades, with borderline being the most common event. And indeed, in the recent study that I showed you, the majority of the diagnosis, whether it was surveillance or for cause, was really a borderline diagnosis defined as I greater than 0 and T greater than 0. In other words, the original 97 criteria. And when we look at the incidence of for cause, they have a bigger distribution of 1A1B and 2A as compared to the surveillance biopsies. Now, I just want to go back to the 93 classification. The threshold set between BAMF borderlines and grade 1, between BAMF borderline and grade 1A was what was used to define acute rejection in this paper. And if you go back to that paper, what was stated in the paper was that the threshold was set, designed so that the false positive rate of diagnosing rejection should be very low, but I will remind you to look at the days of cyclosporin and Nigarad. And I think the problem is there's a potential for a frequent false negative rate then, if we're using that threshold, depending on the prevalence of the impact of BAMF borderline TCMR, which I've already said in many other studies, is the most common form of diagnosis we're seeing in the days of PAC and FDA. So while we push things away with FK-based therapy, I would argue that we push some of what used to be up here in the higher grades down into the borderline grade. And the question is, does that represent rejection or not? By our molecular mismatch, we tend to support that borderline still is a physiologically driven phenomenon related to age-linked mismatch. Now, when we went to look at the impact of these various clinical outcomes post-transplant, either death-censored or all-cause graft loss, what we found was that the first TCMR had an increased hazard of threefold of having death-censored graft loss and a 1.6 hazard of developing all-cause graft loss. And the important point of this analysis was it took into account the subsequent diagnosis in a time-dependent manner of ABMR. So TCMR was still important even if we took into account ABMR for the cohort. Now, in the sensitivity analysis, we looked at the impact of whether that first TCMR was found on a for-cause for surveillance labs. There's no question those found in for-cause had more impact, again, independent of ABMR, or death-censored all-cause graft loss. And if you look at the first TCMR found in a surveillance biopsy, while it didn't quite reach statistical significance, it still had almost a twofold hazard of being associated with death-censored graft loss, although no longer being associated with all-cause graft loss. When we looked by BAMF grades, we found that BAMF borderline had an increased hazard of developing death-censored graft loss. As did BAMF greater than 1A, and again, these were independent of whether they had EGF or whether they had ABMR. Now, what was interesting about this cohort with our frequency of TCMR by BAMF borderline are higher than 30% was that 50% had an ongoing inflammation when they had follow-up biopsy. We find that as being persistent TCMR for the next 6 months of the first TCMR event, and the median time to that event during the second time was 1.7 months. And we call it a subsequent TCMR if the event either has normal normal biopsy in between the first and the second event, or it occurred after 6 Now, we did then a systematic review and meta-analysis asking how common is it that others have reported persistent TCMR in the context of TAC MPA-based therapy. So all these studies were restricted to TAC MPA-based therapy. And if the initial TCMR was greater or equal to BAM borderline, we saw that the 39% were having a second event within 2 to 9 months of follow-up. And if we were more restricted to say the initial TCMR had to be greater or equal to BAM 1A, again, 39% in aggregate were having a second event of BAM borderline or greater in the next 2 to 9 months, suggesting that if we consider TCMR being relatively frequent and a second event being relatively frequent, then there's potential for considering clinical trials for TCMR and ABMR to improve long-term graft outcomes. So this early TCMR can either go into remission with no more graft inflammation, or it can be persistent in up to almost 40% of our patients. And that is clearly associated with the Nova DSA, as is the early TCMR event, which can then lead to a pathway to chronic active ABMR, or the persistent TCMR can go on to chronic active TCMR, both of which would reduce graft survival. So what do we know about clinical trial designs for the treatment of acute rejection? This goes back to the last consensus definition was in 1995, where they talked about steroid-resistant rejection or successful response to therapy. And just to look at what was defined as success, it was a serum creatinine less than or equal to 110% of the serum creatinine on the day of diagnosis, with a return of the creatinine to the day 0 creatinine level. And they were looking by day 5 of therapy. The problem with this paper, going back to 1998, was a consensus from 95, was these are old studies and definitions were potentially and most likely confounded because there was no distinct diagnosis of TCMR versus DSA ABMR versus a mixed rejection. So we don't know really what we were measuring here as a potential steroid resistance or what was defining successful response. And if we look at the EMA guideline that came out in 2009, for acute rejection, the selected primary endpoint should capture a resolved first biopsy confirmed acute rejection episode, second biopsy confirmed acute rejection episode, and patient and graft survival. But when we talk about resolved first biopsy confirmed acute rejection episode, I would make the point that it should not be confirmed by function alone if the histologic persistence is up to 40%, and the fact that our multivariate analysis is suggesting that these second events actually do impact graft outcome independent of other rejections like ABMR. So this really gave rise to what is currently ongoing, which is an international T-cell mediated rejection working group, which the sponsors are AST, ESOT, TTS, and the BAM Foundation, with subject matter experts, regulatory observers, and patients from both AAKP and the European Kidney Patients Federation. And our goal is to really develop a clinical trial design to evaluate novel therapies to treat TCMR. And this is really, it was really, I think, enlightening to a lot of us as we went back and read the literature that the last drug approved for the treatment of rejection was back in 1998, and that was thymo. And the topics that we're working through is defining a standard of care for acute TCMR. We're looking at elements in the diagnostic criteria to define a response to acute rejection therapy. We're looking at operational time definitions for steroid-resistant, refractory, and persistent TCMR. We're trying to come up with a composite primary endpoint for the treatment of TCMR efficacy failure, and then ultimately getting into some designs. We're about midway through. We're in the topic 4. Preliminary, I think where we're coming to a consensus is that the response to therapy will require both functional and histologic evaluation. What will define resolved resolution, I think, is still up in the air at this point, but we're working on it. In terms of ABMR, I think many people are familiar with this study, which showed that de novo DSA was associated with worse graft outcomes. And what we saw was that after the onset of the de novo DSA, the rate of loss of kidney function was accelerated as compared to before the DSA showed up.
And when we looked at what histologic predictors were in a multivariate model were strongly correlated with poor graft survival, it was CG, and I think many groups have shown this now. And so it really gets to the concept that ongoing inflammation leads to fibrosis and ultimately rising creatinine and proteinuria. But this gave rise to then a paper that we published in Transplantation looking at the EGFR diagnosis of ABMR and the subsequent slope in the first year predicts the 5-year graft survival, and that allowed us to ultimately design the IMAGINE trial where we have active ABMR that's enriched for those patients with chronic active ABMR, and they're being randomized to anti-IL-6 versus placebo, where the FDA has accepted the slope of the EGFR at 12 months as a reasonably likely surrogate endpoint under the IND to be able to get early approval with a requirement that we go on to the clinical endpoint of graft loss. So in summary, I think PPAR and drug toxicity limit the effectiveness of the current standard of care, but not all patients need more potent immunosuppression, which is really where we've been spending a lot of our time trying to develop new drugs for all. I think we need to be a little bit more tailored. The evolution of the definition of PPAR efficacy failure, I think, has evolved into segregating out TCMR and ABMR, and a lot of work done by the ESOT group, working with EMA on this. I think we need to consider whether we need to start thinking about TCMR inclusives of BAMF borderline. I don't know that it's as benign as what we all have been talking about. And then the role of surveillance biopsies to detect these outcomes may be required if we're going to look at trial designs that are looking at efficacy failure. But beyond that, I think we need to think about the utility of renal function and follow-up biopsy as an endpoint in TCMR treatment trials, whether it's EGFR recovery and histologic resolution of TCMR below some threshold, for example, less than BAMF borderline. In terms of the utility of renal function as an endpoint in ABMR treatment trials, I think this is already being accepted by at least some of the regulators as a potential surrogate endpoint for these trials using things like EGFR slope. And I'll stop there and open it up to questions.
So Peter, could you hear people asking questions in the auditorium with the microphone when we were walking around?
Yes, I could, okay. But once they've asked if I can't hear it, maybe you can repeat it for me, Candice.
Okay, so maybe you can take the microphone around, and if you can't hear it, let us know and we'll repeat it. Questions?
Marcus.
With regard to a potential secondary endpoint for treatment of ABMR, has any consideration been given to DSA reduction? I'm talking about full-blown ABMR now with DSA. In that a number of studies have shown that DSA reduction seems to correlate with decreased death-censored graft loss.
Yes, so it's a good question. In our own in the PLAZA study, it's being tracked as a secondary endpoint, looking at titers of antibodies as some quantitative way of looking at it. And this was observed in the pilot phase 2 study that was done by George Bolomig in Austria in collaboration with Clemens in Berlin, where they did see a drop in the DSA MFI, if I remember correctly, but it's not been considered as a primary endpoint. No, not at this point.
Other questions? Martin Nassens.
Thank you, Peter, for a very, very nice presentation. And this afternoon we have been hearing already a lot about TCMR and ABMR, so the full-blown TCMR aspect of PPAR. But nowadays in clinical practice we're more and more seeing, as you also indicated, the borderline change with tacrolimus. I think in prior 90 will also show that currently we see borderline changes, and they also associate with, as you nicely showed, persistence of and repeated repeated inflammation later on. Is there any trend to also include or thinking about including borderline changes as inclusion criteria for clinical trials, as endpoint for clinical trials? Do you have viewpoint on that? Because yesterday we were discussing the thresholds, and it's crucial, because if we would include borderline changes in clinical trials, we have to be even more cautious about how we define the thresholds we were at the advanced meeting.
Yeah, so I think, the discussion we've been having in the TCMR working group is really, I think we all can agree that even borderline inflammation is probably not good for the graft, but if we really want to get into a clinical trial design, do we want to be as aggressive to allow an open door to borderline in the inclusion criteria? I think there's probably a general willingness to consider that in the context of for-cause biopsies, and I think again EMA and ESOT made a good argument to the EMA in that regard in terms of whether we should include surveillance biopsy borderline. I don't know that we need to go there actually in a clinical trial design, only because I think there's a lot more justification for randomizing somebody with borderline clinical dysfunction at the same time into a trial, and you're more likely to be able to observe some improvement in that context. I think the problem is going to be, and this is again, I think, a lack of data, but we are seeing persistence of inflammation as being a problem, and I think the data from Brian's group and the data from others have really supported the concept that if we can't shut things down, and especially I think Alex and Carmen were showing this as related to more of a chronic active TCMR phenotype with IFTA, that this ongoing inflammation really was prognostically poor. So I do think we're going to, and there's an opportunity then in using follow-up biopsies to include it in the endpoint, that we will be able to develop new drugs and novel drugs that could treat these inflammatory events. So in terms of inclusion, I would say yes, probably borderline, but only in the clinical for cause context. I don't think there's a need necessarily to go into the subclinical, and I think the ongoing inflammation, we're working on that through our working group, but I think there's a general sense that we do have to see some resolution of this inflammation if we're going to impact long-term outcomes.
The last one.
Thank you, Peter, for a beautiful presentation. I'm actually going to draw from the content presented in your slides and work that you've been advocating for, so the incorporation of the molecular mismatch. In the contents of informing, so to speak, the pre-test probability to complement, supplement, to further facilitate risk stratification of these patients, I just wonder if the borderline findings in combination with significant molecular mismatch would prompt, even in the context of surveillance biopsy, to consider those outcomes. So again, that potentially contributes or lends further support to an immune-mediated injury when observed.
Yeah, no, I think that's a great point in terms of can we use that as a sub-selection criteria for the ones that are more likely to persist or those that are at a higher risk of progression. Yeah, totally good point. I would actually argue that even in the de novo trials, maybe we can use it to enrich for who we really should be trying to enroll into prevention trials. To say these are the patients who are more likely to have a severe event, we should really be putting them, developing new drugs predominantly for them, and not trying to increase the intensity of immunosuppression in a low molecular mismatch group, A, because they're not likely to have an event, and B, they don't necessarily need it in the context of a trial. So I think both ways you could think about how you might use it, either to enrich a prevention trial or be more selective in your treatment trial?
Thank you, Pierre. That's all we have time for. Thank you very much for joining us remotely, despite your new busy schedule.
Thanks a lot, Candice.
So we're going to take a break. It's past, so 15 minutes. We'll start at